quick shout out to all the people listed up here, especially Brad. Thank you for coming. Everything we know about Didymo so far is because of him, and this was kind of his brainchild that we took. So yeah, thank you to Brad, and thank you to everyone on here. And like Bobby said, I looked into the causes of Didymo blooms.
Oh, I was gonna see. Turn on the video. Yeah, why not?
Oh, okay. Okay.
It's not for that.
Are you advancing?
Okay.
All right. So starting with the big picture and climate change, Bobby, like she mentioned, has been up here for 48 years, and 46 of those she has light and temperature data and data on the macroinvertebrates. And so that's pretty phenomenal, but also it's a key into seeing the true patterns and changes that are occurring with our climate. And some of the big four are lower snow melt overall, worse winters, warmer summers, which leads to lower flow, and then also earlier peak flow. And like mentioned a little bit before, with this lower flow, we have warmer water temperatures. So one of the big observable changes that we're seeing in the streams with climate changes occurring is Didymo. And Didymo, also known as rock snot due to its lovely appearance, is a nuisance diatom that grows in freshwater streams. And it also grows in nutrient limited conditions, contrary to most algae. And this is a great graph showing its presence globally, and actually contrary to what you might think of it being nuisance, it's actually native to the area. We have data or like collections of its cells from 70 years ago. And it's not until now that it's started to produce these stalks. So one of the big starting questions is what all of a sudden in 2006 is when we first started to see it form like this in these streams. What is causing this all of a sudden? And what is changing to contribute to this growth? So here's a picture of what the stalks look like under a microscope, and they're actually polysaccharide stalks. And yeah. So the objective of our study was to explore 3 physical variables that we thought could contribute to Didymo's blooming: light, hydrology, so low or high flow, and then also temperature and the interactions between their effects. And we had 2 approaches to our research: an analysis of the long-term data set, along with an experiment. So looking to our first approach, we analyzed the long-term data set. Like said before, we have 46 years of the macroinvertebrates, but it wasn't until 2013 that we got Didymo data. So from 2013 to 2020, we have Didymo biomass indexes from all of the 8 sites, along with light and temperature data from the Hobo data loggers that we install at all of the 8 sites at the beginning of the season. And then we also have this incredible resource down valley, the USGS gauging station on the East River in Almont. And it's pretty incredible. It's been recording flow data every 15 minutes since 1911. So you can just imagine the data set that's there. Looking at hydrology, we have 2 different variables we're looking at, starting with magnitude. So the total amount of water that flows through the gauging station every water year. So down here you have the total amount of water that flows through, and then up here you have the Didymo biomass. And as you can see, we found that as you have less water, more Didymo, higher water, less Didymo. So we also looked at another variable, timing of the peak flow. And we used a variable called QT50, which is looking at the day. So these are all days, number of days. So the day at which 50% of that total flow flows through the gauging station. And as you can see, the earlier that day is, the higher the Didymo biomass, and the later that day is, the less the Didymo biomass is. We also looked into temperature, which is an interesting relationship. This is a great partial plot created by Maraid, and all of these dots represent a year and a site. So there's 64 of these dots. And as you can see, as the temperature rises, so does the Didymo biomass. And that makes sense. And one thing we can contribute, we think is contributing to that effect is its relationship to flow. Flow and temperature, as shown us by this graph, are interrelated and you can't really separate their effects. You would have to do that experimentally. So as you can see, the lower the amount of water, the higher the temperature, and the more amount of water that flows through, the colder the water is. So those are interrelated. Then we looked on to our next variable, which is light, and got a very different result. There is absolutely no relationship or correlation between light and Didymo biomass on the macro level scale, which we were interested by. Which leads us right into our second approach of research, which is our shade cross scour experiment that we set up in Copper Creek. And you might be thinking, well, why test light when you just saw that graph that had no relationship at all on the macro level? And it's because we started looking at the site level and the heterogeneity that is in the sites. So Didymo biomass isn't uniformly distributed at the site level. So as you can kind of see in this photo, we started to observe it specifically at our Copper Creek site that you can see here. There's more Didymo on this side with lower water and also a little bit more light. In the middle, the rocks are lighter because it's higher, possibly higher flow in the middle. And then specifically with Copper Creek, we were looking at the North Bank and the South Bank, and the North Bank has significantly higher amounts of light, like more consistent light, and also has more Didymo. And then on the other side, the South Bank, which is shaded by all the riparian big trees on one side, had less Didymo. So from anecdotal evidence, we kind of justified asking this question. So going into our methods, we set up our experiment on the North Bank of Copper Creek because that's where there's more light consistently. And we had 12 plots, 6 of which we randomized were shaded. And inside of each plot, those black dots are hobo data loggers, which log the temperature and the light that reaches the stream bed every hour. And then in each plot, this is kind of a blow up of one of those plots, we had 2 patches of rocks. And it was either a patch upstream or downstream of the central hobo data logger. And we randomized which patch I scrubbed to simulate scour. So in this one, downstream was the scour and upstream was the control. Here's a picture of the beautiful little tents that Steve, our project engineer, engineered. They're pretty incredible. They're just made out of PVC pipe, and we put rebars on all 4 corners, and then we put this awesome black plastic on the top. And what's cool about it is it's white on the top and black on the bottom, so it didn't absorb so much sunlight and create hotter conditions. And then you can also see we delineated the open plots with rebar and put caution tape around them. Yes. And then all of these central posts have little hobo data loggers that we attached with zip ties and put all the way down on the rebar, so it would collect data at the bottom of the stream bed where the rocks are. Here's an amazing drone shot that someone got of our site and sent to us. It shows our reach, which is actually, it was 70 meters. I did that for you, Billy Barr, because when you came, you were impressed by the magnitude of it. And it actually, yeah, it was a pretty big area. And I also love this photo because it shows just what I was talking about the shade on the South Bank. You can clearly see this shade on the South Bank and more light creeping in on the north. And as the day progresses, it gets more and more light on the North Bank. And so moving on to how we collected our data for the experiment, we used this awesome tool that Brad developed called the Aquagoogie. You didn't develop it? Bobby said you developed it.
He developed it. What?
He's shaking his head.
Who invented the Aquagoogie?
Chester?
Steve?
Well,
I don't know.
well, anyway, someone invented it and it's great. And pretty much what it is it's
Bernard.
Bernard Scotcher.
Oh, well, anyway, it's kind of like goggles. So you place it on the water and you can see clearly to the bottom. And we actually gridded it with tape for this experiment, and there were 32 squares. And what Bobby would do is she would go into the plot, and for each patch upstream and downstream, she would put the aqua gooey against the central rebar and kind of wrap her pinky around the central rebar to like make it consistent every week. And she would count the number of squares to estimate the percent cover. And then she would also measure with a little ruler the height of the Didymos in millimeters. And we did that every week from week 0 to week 5 in all 12 of the plots. Here is some more photos of just the process. We would unfold half the shades and Bobby would go in there and do it. And then we wanted to make sure that we were actually like accurately, like we were efficient, or what's the word? We achieved shade. We achieved actual shade, and that it made a real difference between the patches. So here you can see these were the open and these were the shade, and there was significantly higher light in the open patches, actually 3 orders of magnitude larger. And then we also found, interestingly enough, that also the shaded patches were colder, a little colder too, or a little colder, which we thought was interesting because the water moves so fast and there's such high turbidity, you would think it wouldn't make such a difference, but it did. So that was interesting. And then Brad came and measured PAR in all of our 12 plots, and he found the same thing. He just affirmed that yes, PAR, which is photosynthetic active radiation, was higher in the open than the shaded. And actually you can see here, Steve designed the tents, so it kind of simulated more natural shading. It wasn't like a total blackout. You can see some light can get in, it's just like a little bit of shade. So moving to our results. So looking at this graph, you have on the bottom the open, so all of the different treatments. So you have open control, open scrubbed here, shade control, and shade scrubbed. And then on the Y, you have Didymo biomass index, and we derive the Didymo biomass index by doing percent cover times the height. And this graph is pretty amazing. It shows the average Didymo in all of these plots, treatment plots, from week 0 to week 5. And as you can see in the open control, from week 0 to week 5, there was significant growth compared to look at the shaded ones, and from week 0 to week 5, there's not much difference, and the arrow bars actually overlap, so there isn't a significant difference. And then also in the open scrubbed, it wasn't until week 5 that it started to take off in terms of growth. And this is an amazing box and whisker plot done by Mairead, and the black lines are the medians. And as you can see from this graph, there was way more variability in the data from in the light plots as the boxes are bigger. And you can also see just from this graph that there was higher Didymo biomass indexes in the open plots compared to the shaded plots. And Mairead actually did a ANOVA statistical analysis too on the data, and found that actually light only had the significant effect. Scrubbing and scouring didn't have a significant effect, which was interesting. So some summary and conclusions. So hydrology, looking back to our historical data, was definitely an explanation for variation in Didymo among years, and temperature along with that, with a similar pattern. And those, as I said, are interrelated. And then we also found experimentally that light does explain some heterogeneity within a site level scale. And we're still trying to figure out some possible mechanisms for this. Why are these effects? Why is light affecting Didymo? Why is temperature? Why is the scour? And when I was doing some of my research for my proposal, a lot of people attributed its stock production to this process called photosynthetic overflow. And we're not sure if it's a complete result or cause for stock production, but it could be. And that's when the nutrient limited conditions limits cell division, and then the stalks grow because of the photosynthesis still occurring and that the carbon fixation to create those polysaccharide stalks. And then also looking at peak flow turbidity in light. When peak flow occurs earlier, so does the turbid sediment upturn. So in those earlier conditions where light is also limited because the turbidity in the sediment inhibits light towards the bottom of the stream, Didymo hasn't been blooming yet. So it totally misses Didymo, and possibly that could be what was keeping Didymo under wraps for all these years, is the later peak flow. Didymo would bloom a little bit, peak flow and turbidity would happen and just scour it. And as we saw on the graphs, it wasn't until week 5 that Didymo could pick back up after being scrubbed. And then also, as you can see in that picture, is these huge sedimentation events that we've been seeing as a result of extreme weather patterns happening. So the huge monsoon season, and that limits light a ton, and also impacts just the whole benthetic health. And then also perhaps there's a correlation between temperature and Didymo cell division. And we're looking forward to Brad's looking into some new secret hypotheses that hopefully will reveal some of these mechanisms in a bigger light. So now I'll move on to the so what. Why does more Didymo matter? Why do these blooms matter? What are these changing about the benthetic communities? And Reid will take it from there.